Assalamu alaikum guys, and welcome to my YouTube channel Biology Articles and MCQs. Today we are going to start the topic that is named as counter current multiplier. Counter current multiplier is related to our previous topics. We were previously studying nephron, which is a structural and functional unit of kidney. So the nephron is working on the mechanism of counter current multiplier or counter current multiplier mechanism. Today in this video we will discuss how this counter current multiplier mechanism works. First of all, there is definition that counter current multiplier system is a mechanism that expends energy to create a concentration gradient. Means it is a special type of mechanism in which we will create a concentration gradient. Means in one part of the nephron, or mainly this mechanism is concerned with the loop of Henle. Then in one limb of loop of Henle, the concentration will be higher, and in other limb of loop of Henle, the concentration of the salts will be lower. So there will be a concentration gradient, because in one part the concentration will be higher as compared to the other part, and at the same time this concentration gradient will be created by the use of energy, and energy, it is understood that energy that will be used will be ATP. So let's start that how this all mechanism will work. Here you are looking at the simple nephron structure, and the main points will be concerned with the loop of Henle. This limb is the descending loop of Henle, and this limb is the ascending loop of Henle, or ascending limb of loop of Henle. You know that in the descending limb of loop of Henle, there is reabsorption of water, not reabsorption of salts. The water is reabsorbed because of the presence of special proteins that are aquaporin 1. Because this limb, descending limb, has special proteins aquaporin 1, and these proteins help in the reabsorption of water. So this limb will reabsorb water. Let's suppose that the filtrate that enters into the Bowman's capsule have 300 molecules of salt. Means it may be sodium or chlorine, it may be urea. We are supposing that while entering into the Bowman's capsule, there are 300 molecules. Here it is passing through the proximal convoluted tubules. They are 300 molecules because there is no reabsorption of salts, or the reabsorption of salts is not concerned with the proximal convoluted tubules. So they are maintained a number of 300 molecules. While passing through the descending limb of loop of Henle, there is increasing in number of salts, because the water is reabsorbed, and at the same time sodium and chlorine that is present in the interstitial fluid. Interstitial fluid is the main fluid that is present outside the nephron, outside the structure of nephron. Here the water is entering into the capillaries, and the salts are re-entering into the descending limb. For example, first of all there were 300 molecules of salt, then 100 molecules entered there, and there are 400. At the same time, the filtrate is passing through the descending limb, the water is reabsorbing, and at the same time the salts are entering into the descending limb. Here you are seeing that the number of salt is increasing. The number of molecules of salt is increasing from let's suppose from 400 to 600, and then 600 to 900, and at the same time the water is moving out of the limb. As the number of molecules of salt is increasing in the descending limb of loop of Henle, and the water is decreasing, so there will be the concentration. There will be the concentration that will be the higher. Means there will be the production of hypertonic solution. Hypertonic solution means the number of salts is greater than the number of or amount of water inside the descending limb. So there the concentration is higher. Now moving down the descending limb of loop of Henle, there is further salt increasing, and there are let's suppose that there are not total 1200 number of salts, molecules of salt. Here from the because of the presence of aquaporine molecules, the water was moving out. But when the filtrate from descending limb of loop of Henle is moving towards the ascending limb of loop of Henle, remember this thing that there is no aquaporine proteins are present in the ascending. Aquaporine is special type of protein which helps in the movement of water. But as there is no aquaporine protein present in the ascending, so the water cannot be moved out or cannot be reabsorbed from the ascending limb of loop of Henle. So you can see here that in this portion of ascending limb there is a thin portion, that is thin ascending limb of loop of Henle, and the upper portion is thick ascending limb of loop of Henle. There is the difference between these two thicknesses, and at the same time also they differ also on the basis of the presence of sodium channels. In the thin portion of the ascending loop of Henle, there are simple sodium channels present, and these sodium channels, as its name shows, will help in the passive movement of the sodium from higher concentration to lower concentration. As here there are 1200 molecules, that is higher concentration of sodium, and some of the molecules will move out into the interstitial part. Then the remaining sodium molecules are, let's suppose there are 700. As they are moving from their high concentration to lower concentration, there will be no use of energy. They are moving passively, as that of water molecule that are passively moving from the descending limb from its higher concentration to lower concentration. Let's suppose some of the molecules have moved out from the ascending limb, or thin portion of ascending limb, and there are now 700 salt molecules remaining in the thin portion of ascending limb. Now you can see here, let's suppose the 500 molecules of salt or 500 molecules of sodium is moved out from there, from 1200 molecules, and the remaining molecules are 700. Now from 700 further 300 molecules move out, and the remaining molecules are 400 in the ascending limb of loop of Henle. You can see here first now they are in the interstitial fluid, there are total 800 molecules of salt. 500 plus 300 will be 800, and but now in the ascending limb of loop of Henle, in the thick portion of ascending limb of loop of Henle, there are only 400 molecules, that is less than total 800 molecules of sodium or chloride or simple salts that are present outside the ascending limb. Now there is higher concentration outside the ascending limb and lower concentration of salt in the ascending limb of loop of Henle, in the thick portion of ascending limb of loop of Henle. Now but the main point is we need these 400 molecules of salt also for various metabolic processes that are carrying out in our body. So in order to move these 400 molecules outside the ascending limb, there will be automatically use of energy, because this movement will be from the lower concentration to higher concentration. The use of energy and this energy comes from the sodium potassium ATPase pumps. These ATPase pumps, sodium potassium ATPase pumps, will use ATP energy energy in the form of ATP, and then move these molecules also to outside the ascending limb of loop of Henle. So let's suppose that first of all 200 molecules removed and remaining are also 200, then 100 molecules from 200 molecules removed, and the remaining molecules of salt are now 100. And this movement is from lower concentration to higher concentration, so it is using energy, and energy is coming from sodium potassium ATPase pumps. So in this way you can see that there will be the higher concentration of solute or high concentration of salts in the descending limb of loop of Henle, and there is a lower concentration of solute or lower concentration of salts in the ascending limb of loop of Henle. As there is created a concentration gradient, here is higher concentration and here is low concentration gradient, so this mechanism is counter current multiplier mechanism. Counter current means the opposite gradients in the two limbs or in the two different parts of body, or you can say in which part of body you are studying the counter current mechanism. Some important points about counter current mechanism are that the counter current mechanisms works only in the portions where there is hairpin loop like structures, means it is a hairpin loop like the hairpin you wear on your head is in the same way as these both loops are present. And the second most important point about the counter current multiplier mechanism, that it works in those parts in which the movement of solute or movement of fluid will be opposite in both parts. Means here the fluid is moving upside down, and in the second portion the solute is moving or the fluid is moving from downside to upside. So this is all about counter current multiplier mechanism that is running in our nephron. This counter current multiplier mechanism is important because here we conserve most of the amount of water and most of the amount of salt which our body need in the daily processes. Here most of the amount of salt will be conserved, and the remaining amount of solutes, means that is urea or waste product which we don't need, will move to the collecting duct and it will be removed out from our body in the form of urine. I hope you understand all the topic. If you have any problem regarding in this topic, you can tell us in the comment section below. Thank you so much.